Humanized models are also being developed by scientists basically to address a lot of therapeutics nowadays. So we have different types of animal models. So one is spontaneous models. For example, this is indoor mice. You can see here. There is a spontaneous model. It was not genetically modified. So over a period of time, the inbreeding has taken place, and that animal is a good model because for cancer biology, they do not have a thymus gland. So that's why T lymphocytes will not be there. So hence rejection will be very less in these animals. It's a good model for. And other than that, so we also have a lot of other animals which then do directly get same kind of disease. What, for example, cat get asthma. So the cat is a good model for asthmatic studies in human beings. And again, iris setter dog. So if the factor 8 deficiency is there, so they are good model for human. Inducing obesity in animal it takes a lot of time. Rather, if you knock out the gene particular, and you can induce the obesity very fast, and then you can use it for experimentation. So again, selection of animal models is very, very important in biomedical research. So there are various type of animal models available. So scientists has to do a lot of homework. For example, so in veterinary colleges or in pharmacy colleges, most of the time we use ceftriaxone or Alexa to induce diabetes. But the problem is just whether it's a good model or not, we have to question, because Alexa induces diabetes within 48 hours. All the beta cells will get destroyed. Whereas in humans, we don't get similar kind of disease progression. So it's a slow progressing disease in human beings. So that's why people prefer now the knockout models, because very knockout models will develop the diabetes slowly compared to people prefer humanized mouse models because it's very easy to use. These are some of the autoimmune diseases, like inflammatory bowel disease. Yeah, rheumatoid arthritis, multiple sclerosis. This is very difficult to induce in conventional strains. So that's the reason. So you can see the various type of models, the transgenic models are being used for this kind of purpose. Again, cancer research is another major area wherein a lot of people are working. So we have a lot of transgenic models for these kind of things. Again, depending upon the availability and the cost, so they can go for it. These are some of the important transplantable tumor models, genetically engineered mouse models, and again carcinogen-induced tumor models. So various types are there. So scientist has to select whichever is appropriate for him. Again, so the great advantage of these genetically modified or genetically engineered tumor models is it's possible to model tumor development and pre-malignant neoplastic stages in animal, so more or less similar to human beings. Again, tumor heterogeneity will be there. Tumor microenvironment is almost more or less similar. Again, studies on defined mutations is also possible by using these kind of animals. These are some of the important subcutaneous models for cancer biology. So these are the different cell lines, and you can see what kind of cancer we are getting. So and that these are the cell lines we use, and you can induce the tumor models. So similarly, for orthotopic models, we have a lot of cell lines for that. And again, carcinogen-induced, there are actually chemicals also we can use, like dimethyl benzoanthracene also is one of the commonly used chemical for inducing skin papillomas. But they are not so good model, but people are using genetically engineered animals nowadays, because publication in a peer-reviewed journal is very difficult if you use the olden techniques or the animals, the conventional animals. So again, humanized mouse model is getting a lot of importance nowadays. Basically, the immunodeficient mice is engrafted with human hematopoietic cells or tissues, so that transgenically express the human genes over a period of time. So this is the latest development. So people are using, and it's very difficult to maintain because both T and B lymphocytes will not be there. They are highly immunocompromised, and maintaining them under the highly sterile condition is required. Otherwise, even if one bacteria or virus enters into the body, animal will die subsequently. So that's the reason. So even the maintenance also is very, very tough job in case of these mouse models. Some of the immunodeficient mouse what we are using nowadays for cancer biology is NSG mouse. This is actually most of the facilities in India are started using this NSG mouse. So this is the best model for cancer studies. So if the scientist has to select, so the best model, if he is using some, for example, cardiovascular research, if he is doing, so he has to select some of the models depending upon his research interest. So similarly for CNS diseases and diabetes, and for metabolic and regulatory studies. So these are the transgenic models. You see, nowadays most people are turning towards the transgenic models because it has a lot of advantages. Again, immunology models, So oncology models, these are some of the examples. I will just quickly go through it. And so, unfortunately, so most of these transgenic animals are not available in India. We have to import it from these 4 important sources, international vendors. So they do give to India, but so we can breed and we can use it for our own purpose. But they are very expensive to buy and also to maintain also sometimes, genetically modified animals. Next one is the communicable diseases. So actually, I think you're all aware. So we are using a lot of animals for challenge studies. So there are different categories of biosafety, BSL1, 2, BSL4. So we have a couple of facilities in India. So the problem in conventional animal is they do have innate immunity. Sometimes they may not respond to the challenge studies. So that's why. So here also we use a lot of transgenic animals for inducing or challenging with a particular bacteria or the virus. So here recently NCBS during COVID time, so NCBS also has developed 2 models which are very famous. They have developed transgenic animals to understand the therapeutic or vaccine effect on the COVID virus. So similarly, monkeys are also being used for AIDS, but the problem is it's very difficult to induce AIDS in monkeys. They are only susceptible for simian immunodeficiency virus, not the human immunodeficiency virus. So, but these 2 species are susceptible. Hence, but the problem is they are not available in India, and also a lot of ethical issues are there with this one. But still, some countries are using this one, still doing AIDS related work in this species. So next important category is preclinical research. So almost 70 to 80% of the animals what we are using in India are under this category. So in India, approximately around 20 lakh animals we are using every year for biomedical research. So out of that, around more than 15 lakh goes only in this sector, preclinical research. And this is actually basically it's a vital phase in drug development where potential new treatments are tested in the lab-based. We have both in vitro and in vivo models. So before the human trial, it's a regulatory requirements. First, you have to test it on animals. Then only you have to submit the data to the regulatory agencies. There are various regulatory agencies across the world. So this stage is very critical for gathering the essential data on the drug safety, efficacy, and also to determine the dosage in human beings. So that's why animal experimentation is very, very essential. Basically, safety and the efficacy. So nowadays, a lot of in vitro tests are also available, but regulators require mostly the animal safety data. So it's a regulatory discovery process is a very complex procedure, and it's a time taking. So it will take 10 to 15 years minimum if the drug is synthesized to come into market. So we use a lot of animals in this stage, preclinical stage. So it takes 1 to 2 years because there are some studies like reproductive toxicity studies or carcinogenicity studies that will take minimum 1 to 2 years. So here we use a lot of animals, and then once it is passed in this stage, then it will go to clinical trial. So this is just an estimation. So day in and out, scientists are synthesizing several thousands of chemicals, but out of that, so they will get eliminated in the preclinical stages itself. Out of 10,000, so almost only 250 will get passed through this stage. So that means animals are basically the filters for all these chemicals, so which are toxic in nature. So once it passes clinical stage, preclinical, then it will go to clinical stage. Out of 250, only 5 will come to human trial. That's all. So you can see how we are playing a crucial role in filtering or checking the efficacy of these drugs for human use or animal use. So there are a lot of safety studies are there. There are a lot of regulatory guidelines are there. So again, the regulatory authorities basically request 2 species because extrapolating the data from 1 species is sometimes very difficult because rat is not similar to human beings. So sometimes you may have to use dog or monkeys depending upon the intended use of that particular compound. So 2 species is required, but in some cases in life-threatening diseases, so fewer studies to reach clinic because in preclinical studies there are a battery of tests. OECD has around 150 different type of tests are there. So at least you have to go through some 15 to 20 different type of tests before it goes to human trial. Whereas in case of emergency, so some of them are allowed, like in COVID time. So COVID vaccine came to market within 6 months. Actually, it should have taken at least 1 to 2 years to check the safety of the particular vaccine. But somehow it was emergency in use was permitted by the government. So hence, so some of the rules can be. circumvented depending upon the reason. So we have a lot of toxicology models. Most of the drugs, depending upon the intended use, it has to follow the different guidelines what we have. So we have different guidelines by the OECD. So most pharmaceutical companies, so they follow these OECD guidelines, because once you pass it through these guidelines, or that animals are used for these kind of studies, then it will go to human trial. And again, we also have a national guidelines, CDSCO. So their limited guidelines are available. But if the drug is intended for Indian usage, then you can follow these guidelines. If it's for international, OECD guidelines are required. So we have around 150 different type of guidelines are there. Out of that, around 70 uses animals, different type of acute toxicity, subacute toxicity, chronic toxicity, reported toxicity, various type of tests are there. So under these 5 sections are there. Under that, biotic system and health effects use a lot of animals actually. So these are some of the guidelines what we use for testing the safety and efficacy of the compounds on the animals. So we use fish, birds. These are some of the images I have taken. So most commonly used is zebrafish, and then we also use birds. And again, for biotic system, we also use different type of insects, honeybee, earthworm. So basically, if the compound is toxic to the environment, then it has to be tested on these kind of animals, basically to avoid environmental contamination. So these are some of the test systems. We call it as a test system rather than the animals. So in case of laboratory animals, we use rat, mouse, dog, hamsters, hen, everything. And you are all aware that in India, so we have the regulation for animal experimentation. CCCA basically controls all the experimentation. We have more than 1,600 animal facilities registered with the CCCA, and out of that, majority of them, almost 70% of them are small animal facilities. Very few large animal facilities are there, and this is just a graphical representation of the different facilities. And we do use large animals for biomedical research, like cattle, sheep, goat, camel, horse. And in India, currently we are using more than 10,000 horses in the research field, basically for production of anti-snake venom and ATS. So maximum horse is being used in this sector. And apart from that, we also use mini pigs and monkeys, dogs, for experimentation. In India, so we have only 3 species, but in the world, there are 30 species of monkeys are being used for biomedical research. But in India, so we don't have that much. Only 3 species are available, and these are the places where we are maintaining the primates for biomedical research. These are commonly being used primates in India. So, and this is one setup. Just I want to show you how advanced we are in case of biomedical research. This is not a human hospital. This is a dedicated cath lab for the dog and the pig, sheep. So this is in Hyderabad. We have 3 such kind of facilities in India. So it's amazing to know that this kind of facility we are using for biomedical device testing. Basically, this facility is involved in testing the biomedical devices like cardiac stents and the biodegradable stents and drug-eluting stents, and so many other biomedical devices they are using by using pigs and the sheep. And the compound which are tested in this facility directly goes to US FDA approval. So this is one of the latest facility what we have in Hyderabad. And so, and also, so I'm very proud that our veterinarians are able to see treating the animal is very easy. But here they create a situation like hole in the heart, or they create aneurysm, or they create a stenosis, and then they use the device to check their efficacy of the particular device. So that's a very complicated procedure. First, creating the disease condition in this model, large animal model, is a tough job. But so veterinarians are doing this one. So really. So we should be proud of them. And in addition to that, we have a lot of BSL-3 facilities also in India. So earlier, so we had very few number. Now after the corona, so we have increased. Earlier it was only 18 labs. Now around 100 plus labs are in progress. I think probably in another 3-4 years, India will have more than 100 BSL-3 facilities, and some 4 to 5 facilities BSL-4 also. So we have some limitations with animal models. One is sometimes inaccuracy in prediction, because if you are using improper model, and inability to replicate complex human disease sometimes, like cancer and Alzheimer's disease. and ethical concerns also sometimes. So we are always under constant pressure from the animal activists for dogs and monkeys if we are using. So that's also one big stress for the scientists who are using large animals, and high cost. And again, limited availability of certain animal models. Right now, so very few facilities are breeding good quality animals. We are spending around 100 to 150 crores every year to procure only the animals from abroad. And also, I would like to mention here this preclinical study research sector has generated 10,000 crores last year. So that's one big achievement so we have made last year. And so in future, what we have to do is we have to develop more complex animal models. People are developing, and again advances in gene editing, and again 3D printing and tissues organs are future directions. And I want to mention here is more than 75 Nobel Prizes in 110 years awarded to scientists who have used animal as a model system. So this itself will speak the contribution of animals to the human health and the animal health. So this is the greatest contribution. This is some of the list of the things I have taken from the web. So these are the list of scientists and the animal which they have used for their research. So this big list is there, and the important milestones what we have achieved in the last one decade, one century. By using animals is first the development of local anesthetics, or development of heart-lung machine, or even kidney dialysis unit. So and again hip replacement surgery. All these things was possible only with the help of animals. So their contribution is really great to the biomedical research. Yeah, in conclusion, animal models have played a crucial role, and but there are limitations, and again animal models that makes them less effective for certain type of research. So scientists do agree, but sometimes we do not have any options. So sometimes we override certain things. Again, development of new models are required to address a lot of emerging diseases. Again, humanized mouse, and even now organ on chips have come. So to avoid all this animal usage, so a lot of research activities going on. So just on the slides, they prepare all these organs and then they use it as a in vitro model. So basically, 3D printing of tissues, they call it as. Yes, that's all.
Thank you, Dr. Anthony.
Yeah.
Very wonderful. And I can say very thought-provoking, wonderful presentation. Thank you very much. And I beg pardon from the prayers and the delegates that I have to meet. So after receiving it, I think my co-chair will take care of the session. Thank you. Thank you.